Hello. Thank you for the introduction. Actually, I'm going to talk about deep learning and cats. That's very interesting because there's been quite a bit of work going on lately with LLMs trying to compare them to cats, hence the quote that is here. And it turns out that cats are actually pretty good models of deep learning networks for a couple of reasons. The first one is cats have about the same amount of neural connections than the best language models have today, so kind of similar in size. The second thing is that cats have both prey and predators, so they are imputation machines. If you have ever seen a cat play or being activated, it just tries to predict everything. So that's perfect because that's exactly what the neural network is trying to do as well. And the third thing is that cats are very biased in their training. Just like neural networks, they make a lot of mistakes based on the information that they get during their training, and so they make a pretty good model. And to demonstrate this and kind of give you an idea of how that might imply to what we are doing, I ran a bit of an experiment. So here is a cat, actually, and I have in my hand a cat snack. And what I did is I trained this cat to run after the snack. So I just throw snacks, right, for quite a while, and then the cat just runs after it gets it and then comes back to ask for more. And then at some point, that was a training, and at some point I decided I'm going to stop to do that, and I'm going to fake throw, right? I'm going to just make the movement, but not let the snack go. So we can actually measure the ability of the cat to generalize to its training. So on the y-axis here, you have the distance traveled in meters, and on the x-axis, you have the number of fake throws. Actually, in the control, the cat goes to the end of the room, which is here about 4 to 5 meters. And after I perform a couple of fake throws, we can see that the cat is still looking for the snack in the same place. After a little while, it starts to understand that something wrong is going on. There is no snack at the end of the room. And yes, at the end, it just stays there and like, I'm not going to move. And then the interesting question becomes, what happens at that stage? What happens is, at this stage, you start to throw another cat snack. So I'm going to demonstrate this live. I have a cat snack in my hand here. I'm going to throw it. Okay. You hear that the sound of the snack, it goes after the hand movement. And what happens is that if you throw it, the cat doesn't move straight away. It waits for the sound cue. And that's very important for what's going to happen next. So let's formalize the problem a little bit. So we have a cat here. At the top, you see the cat symbol. And it's trying to find out from its environment, that's an X value, that's an input, if there's a snack or not. During the beginning, we are doing only real throws, which means that the hand movement is perfectly correlated with the presence of the snack. And it's going to be the learned predictive feature, because just as I demonstrated, the movement comes before the sound. So it's a much better predictive feature, which means that the cat gets to the snack much faster. During real throws, however, so fake throws and real throws, this is not the best predictive features anymore, because it is not perfectly correlated. But the cat is very smart. This is how the comparison to deep learning comes in, right? The cat is very smart. Very quickly it understands that it needs to change the way it processes features. Deep neural network, on the other hand, are not very smart in that regard, because we've trained them, and after we have trained them, we freeze them. That means that we're never going to change them again. And so there's a bit of an impenetrable wall between the training and the testing. So if the neural network would be a cat, it would always run after the invisible snack, and it will break, essentially. So our work here is to kind of look at the train and the test, because this is where most of the things happen. And comparatively to animals, in deep learning models, we have to get this part extremely right. So let's kind of look at some arrays of tasks that we do with sequence in deep learning. One of them could be like DNA sequence transcription, DNA sequence transcription factor binding, or you can predict also structures. And in all of those fields, we have misleading features, just like our fake hand movement here. And I can develop one of those, for example, absolute position bias. If you are training a deep learning model to predict DNA transcription from DNA sequence, some people will align all of the sequences. It means like you have this anchor here, and all of the sequence will be aligned on that anchor. That means that the model will rely on the absolute position to predict the value. So at position +2, I have something interesting. And what if in the real world your things are not aligned anymore, and then it will break? So that's one of the examples of the fake stuff going on there. And a good question from that point is, how do we solve this problem in a deep learning for life science setting? Which means that, in that case, we have no idea what are misleading features. We have no idea what are the hand movement and the cat snacks. What do we do in that case? That we make our cat blind. Right? That might sound a bit brutal, but this is what we do. So what happens is that we take our train set and then we try to noise it in a very weird ways and in a lot of different ways and in ways that are more difficult to recover than others. And then we train a neural network on each of the ways the data has been noised. So each of our noisy networks, they then are going to try to predict on the original datasets and some of other noise datasets. And if we actually get better, that means that at training time, we have removed one of the features that was really bad for our model to learn. So that's a good sign. If we get worse, that means we touched something that was important and we should probably not touch. So this is a project we developed to solve this particular problem using nextflow. Nextflow is extremely useful here because you see this is a problem that is embarrassingly parallel. So we have a lot of data set that we can launch and a lot of small deep learning jobs that we can launch. And this is really great to be able to use nextflow to scale up all of those jobs. And to test our approach, we participate in a challenge, which is called EBIS. It's for assessment of transcription factor binding affinity. And really quickly, what happens in EBIS is that they are going to give us a bunch of DNA sequence in vivo, so human DNA sequences, that have a check mark. It's like, yes, if the transcription factor binds to it, and no if it doesn't. And our model is going to learn on this. And then they are going to give us another set of sequences that are lab-generated synthetic sequences, and those sequences don't have any label. We have no clue. So we learn the human sequences, and then we try to predict the synthetic sequences, and we send to EBIS the results. And then they send back to us like, yeah, this is, you did good, or you didn't go very good. This is really great for our problem, because if our model is looking at misleading features, so the kind of hand movement I was showing before, it will not be able to generalize to synthetic data. So that's a really good benchmark. So how do we do on those? We decided to start by taking the data and trying to understand what are the possible artifacts or misleading cues that could be in that data. We found two in particular. One of them could be the fact that you have experimental artifacts, which means that the tools that were used to generate it or to parse or to process those DNA sequences in the in vivo set would be different than the one in the in vitro set. So that would mean that we would have some artifacts in our sequence. And if we would noise the sequence by masking randomly some percentage of the nucleotides, we could manage to erase some of the artifacts in some of the sequence just by chance, and that would mean that we will not learn them. And another thing is that the tools that were provided do not make the difference between plus trend and minus trend. So that means that in our data set, the sequence should all have their reverse complement so that the model can find out that, oh, there's a reverse thing. So this is the kind of two noising or transformation process that we imputed in this pipeline. And here are the results. So on the top line here, you have the default model, which is a model that is learned on the vanilla dataset, the one that was given to us. And then if you look at the other lines, these are the models that are learned on the other types of dataset that have been noised and transformed. You can see that the best model is actually not the one trained on the default data, which support our hypothesis. The best model is the one trained on the reverse complement dataset, and that also have 10% of nucleotide masking. So we took that model, because it was the best for us, and we submitted it to EBIS. And what happens is quite interesting, is that on synthetic data, the model that has been trained on all the noise and things that could be a better idea for us, right? The model that was trained on all these things is much better than the default model. So here you have a metric of AUC on the y-axis. You have to know that 50% is random. So the model that is trained only on default, it's a small model, is just a bit better than random. But as soon as you try to add all those noise techniques to try to mask out a bit of the bad features that the model should not learn, you actually get much better. So that's one of the aspects of our pipeline. It's one of the things that we try to do. But we have much bigger vision for that is that we would like Stimulus, our pipeline, to be a hub for all the possible test cases that you can implement on a deep learning network. Because deep learning networks are very difficult to debug, very difficult to understand, and the data that we are training them on is even more difficult to debug and even more difficult to understand. And therefore, we need to have a bunch of test framework in the same test bench that all live together, so that if a user gives a model, they will be able to test that model in a lot of different ways. And some of the interesting ones here are, like, for instance, interpretability, type checking. So some models run on different hardware, and sometimes they use not the same kind of floating. So some use float 16, some use float 32. That have a lot of impact on performance. Some analysis to understand if the model is learning always the same features, and if it can generalize to out-of-distribution datasets. So there's a lot of actually testing that we can do thanks to the advancement of theoretical deep learning in the last couple of years. If you want to build this with us, that would be so much welcome, because this is a lot of work and we are not so many. We have a Slack channel. We are actually trying to put the pipeline into an F-core under the name Deep Model Optim. And on the Slack channel, we are posting a lot of stuff. So it's either code stuff, but we also post vulgarizations of deep learning principles. So if you are just interested to learn, review papers and those kind of things, this is also a nice place to join. And we'll be very happy to have you. So that being said, I would like to thank all of the people that have participated in this project, and the people at Sequoia who developed some of those things with us and that could sit in the meetings, listen to me speak my mumbling, and then the people who actually contributed to the pipeline, and all the people in F-core that made this possible. And it's missing the people in the hackathon over the last couple of days that made this possible as well. Thank you.